Let us understand about how we can perform filter validation. So in sterile manufacturing process, in aseptic manufacturing process, filtering through point to micron filter is very important. It achieves the sterility for the aseptically processed sterile filter products. How we can validate filter? How we can validate or ensure that point to micron filter is able to retain all the bacteria? The test for this is called bacterial retention test. It is as per ASTM F838-20 as well as PDA Technical Report 26. It defines the methods or method how we can validate the filter through bacterial retention test. So let us understand how can we do it. So what we need for the validation? We need culture media with 10⁷ per ml concentration of Brevandomonas diminuta. So this culture media is required. We need a vessel to make media, then filter to be tested, then control filter, two control filters, one is for negative and one is for one is to check whether any bacteria which is coming out of the filter understate to retain that bacteria. So these three filters, test filter and two control filters, and sterilization or sterile workbench. These are the things which we need to perform the bacterial retention test. So how we can start? So we first have to prepare culture media. So the culture of Brevandomonas diminuta ATCC 19146, that has to be culture that has to be inoculated in tryptic soy broth to achieve 10⁷ per ml concentration of the bacteria. This is now going for the challenge organism for the filter. For the purpose of test, we need to sterilize pressure vessel, we need to sterilize tubing, filter housing, and the filters, filter under test and control filters all are has to be sterilized. That has to be assembled under the LF workstation under LF. Now first we'll pass the buffer solution. So we'll fill the vessel with the buffer solution. That sterile buffer solution we have to use. Pass the sterile buffer solution from vessel through tubing to the test filter. Remove the excess air from the housing. Allow the solution to pass. Allow the solution to pass to the two control filters, one negative filter and one control filter. The flow rate we have to keep is 1 liter per minute, and the pressure which has to apply is 30 psi pressure we have to apply it, and 1 liter per minute flow rate has to be achieved. Pass sufficient volume to wet the entire filter, test filter, as well as the negative control filter and the control filter. Once this is done, then incubate the negative filter into the culture media. So negative filter, that to ensure that there are no obviously the organism present into this entire sterile setup. If there are already a bacteria is there, then the test will not be valid. So we have to incubate the negative control to prove that this entire assembly is sterile. Once you pass this saline buffer, now next is my actual test. So what I have to do, I have to take the culture with Brevandomonas diminuta with 10⁷ concentration. We have to pass it through the filter slowly. The flow rate will be 0.002 to 0.004 ml per minute per cm square of the filter surface area. So this low flow rate per the cm square of the surface area of the filter we have to pass, and to ensure that the entire quantity is passed to the filter, and this sterile effluent through the observed test filter goes to the control filter. So any bacteria which is not retained will be retained by the control filter. After this is over, we have to incubate the control filter as well as the test filter. So this is my test. Now how to evaluate the results? So negative control filter should not have any microorganism. The test filter obviously should have this Brevandomonas diminuta, and the control filter which is there to test, to retain the bacteria which are not retained by the test filter, that has to be incubated. So the control filter which is used to challenge the retention capacity of the filter should not have any bacteria, or if the colonies are identified, then it has to be checked for the organism. If it is Pseudomonas diminuta, then we have to calculate the colonies, and basis that log level retention of the filter has to be determined. Practically plus minus 1 log level variation is allowed. So if Pseudomonas diminuta is there and it is meeting the log level reduction criteria of the filter according to 10⁷ challenge organism, then it makes that filter is capable. If the log reduction is more than that, then we can say the filter retention is not effective. Apart from this, if any other organism from the control filter apart from the Pseudomonas diminuta is identified, then it means this test is invalid, because this filter already have a contamination, or the challenge organism already have other, or filter has already have other contamination, so it cannot be considered valid. So just to summarize the entire testing, we need culture media, we need test filter, two control filters. We have to pass the saline solution to weight all the three filters. After weighting, the negative control has to be incubated to ensure that entire assembly is sterile. Then challenge organism 10⁷ Bacteriomonas diminuta culture we have to pass through 0.002 to 0.004 ml per minute per cm square of the filter surface area, and then incubate the test filter and the control filter. Control filter effectively should not have any marker organism, or if it is said, it has to be calculated as per the log reduction. If it is meeting the 10⁷ log reduction criteria, it is passing. If it is not, it is failing. And if we have any other bacteria than the Pseudomonas diminuta, then test is considered invalid. So hope this video makes you a little understanding about how we can do the bacterial retention test as a part of filter validation study. Thank you very much for watching my video.